Cell Cycle Regulation by Martha Ugalde for Concepts of Biochemistry at Grand Canyon University. All right, guys, I'll be talking about the cell cycle regulation. So in my presentation, I'll be doing a brief overview on the cell cycle, explaining the importance of regulation, going over the checkpoints that you see here in the G1 phase and the G2 phase. And of course, there's one right in the middle of mitosis. I'll be going over these checkpoints. I'll be talking about the key regulators involved in cell cycle regulation, the mechanism of regulation, and I'll be providing an example for dysregulation, and then wrapping up. All right, so brief overview of the cell cycle. My laser going here. So eukaryotic cell division proceeds through a structured sequence of stages, beginning with the G1 phase right here. It takes approximately 6 to 12 hours. This is the phase where daughter cells pause before further division takes place, and this happens particularly in rapidly growing tissues. Next, in between, there's a pivotal checkpoint that takes place. It's the R point within the G1, which determines the cell's commitment to continue into the S phase where DNA replication occurs for the newly forming or forthcoming daughter cells. So following this G2 phase, or following this, the G2 phase synthesizes new proteins and cell growth, effectively preparing the cell for mitosis. During the M phase, the nuclear envelope dissolves and chromosomes segregate to the opposite poles. The nuclear envelope begins to form around each set of chromosomes. And then finally, cytokinesis divides the cell into two distinct daughter cells. This entire cycle spans approximately 24 hours in cultured animal cells. So as you can see here, we have the G1 phase takes about 6 to 12 hours. The S phase runs about 6 to 8 hours. And then G2, 3 to 4, and mitosis for 1 hour. So why is it important to regulate the cell cycle? So in eukaryotic cells, signaling pathways are fundamental in controlling the cell cycle, particularly during embryonic growth, where cells divide extensively. In adult tissues, cell division becomes largely inactive, which highlights the importance of regulated division for the organism's health. Failures in these regulatory mechanisms can lead to unchecked or unsupervised cell division and, of course, result in cancer. Proper cell division requires a precise sequence of biochemical events to ensure each daughter cell acquires essential life-sustaining molecules. Universal regulatory mechanisms have been identified across eukaryotic cells that dictate the timing and progression of cell division. Regulation is crucial for organismal development and tissue maintenance as it maintains cellular homeostasis, it prevents uncontrolled cell proliferation, and upholds genetic integrity through accurate DNA replication and repair. It also coordinates tissue development by controlling division differentiation, and growth. And then lastly, it responds to cellular stress by stopping the cycle for repair and preventing premature aging by regulating cellular senescence. Here we have the location of these checkpoints. So from G1 to the S phase, there's right before G1 ends is the first cell growth checkpoint, making sure that everything, of course, that is necessary, it can proceed into DNA replication. And then, of course, we have a DNA synthesis checkpoint. As I said earlier, it's to ensure that the DNA has replicated faithfully and is okay to push forward for the formation of the 2 new cells. And then this checkpoint happens in between metaphase and anaphase, ensuring that chromosomes, sister chromatids, are separated correctly to the opposite poles. So cell cycle checkpoints. These checkpoints are vital mechanisms that regulate and ensure the fidelity of the cell division. The G1 checkpoint located again between the G1 and S phases determines if the cell is prepared for DNA synthesis by assessing DNA integrity, the cell size, and nutrient availability. So pretty much the cell has to be mature before it exits G1 and enters the S phase. If conditions are not favorable, the cell may enter a resting or G0 stage or engage in repair. The G2 checkpoint, situated between the G2 and M phases, checks for DNA damage and verifies that DNA replication has been completed, it is accurate, and ensures that the cell is ready to undergo mitosis. If issues are detected, the cell can attempt to repair it. If the damage is repairable, it will direct the cell to undergo programmed cell death or apoptosis. The M checkpoint, or spindle fiber checkpoint, this one happens during metaphase of mitosis and ensures that chromosomes are correctly aligned to spindle fibers before they're pulled apart to separate them. This checkpoint prevents chromosome missegregation by halting the process until proper attachment has been confirmed by the spindle fibers. Collectively, these checkpoints maintain genomic integrity and facilitate precise division. So here's our structure. All right, very first one at the end of G1, again, ensuring that the cell is the appropriate size, that it has the correct amount of nutrients. Okay, if damage in the DNA is found here, then the cycle will come to a pause. All right, the second checkpoint here is DNA damage checkpoint, ensuring that DNA has replicated faithfully. And when all is well here, it moves on to the next checkpoints. Key regulators of the cell cycle include cyclins and cyclin-dependent kinases, or CDKs. So I'll be going over the importance, super important structure in cell cycle regulation. So the green over here, the green structure is a cyclin, and it is bound to an enzyme that is a cyclin-dependent kinase. This cyclin-bound, cyclin-dependent complex ensures that the cell cycle can progress from one stage to the next. Okay, so cyclins are proteins that regulate the cell cycle's progression by activating the CDKs. Those are the enzymes that drive the cell through the various phases. Cyclins have to bind to CDKs. They form this active complex, and it phosphorylates target proteins, allowing the cell cycle to advance. So a cascade of events get triggered essentially with this cyclin and cyclin-dependent kinase. Cyclin levels also fluctuate throughout the cell cycle, ensuring precise timing of cycle events. Okay, so I have this graph here that represents the importance of this. So between G1 and S phase, we have the cyclin E and CDK2 complex that is abundant. All right, and for the next phase, as you can see, from S phase all the way through G2 phase, okay, just up until the beginning, I want to say about 20% of the mitosis phase, we have cyclin A bound to CDK2 complex that is visible. And then cyclin 8 bound to CDK1 is present only during the mitosis phase. Excuse me. Different cyclin CDK combinations are responsible for transitions between specific phases such as G1 to S, G2 to M. This regulation ensures orderly cell division and prevents errors from happening. It contributes to cellular homeostasis and, of course, genomic integrity. We want to make sure that DNA is faithfully replicated. All right, so in this slide, I'm going to talk about cyclin-dependent kinases. They are pivotal in regulating cell division, and they do this by phosphorylating key proteins. So these proteins modulate essential cellular processes. So during mitosis, these CDKs phosphorylate lamin proteins, and they lead to the depolymerization of lamin filaments and the breakdown of the nuclear envelope. In cytokinesis, CDKs will phosphorylate the regulatory subunit of myosin, causing the dissociation from actin filaments and an activation of the contractile machinery, which is later reassembled for subsequent cell division through dephosphorylation. CDKs also play a crucial role in cell cycle arrest by targeting the retinoblastoma protein, or PRB. So I'll be referring to retinoblastoma with PRB. All right. So unphosphorylated PRB will bind to the transcription factor E2B, and it prevents the transcription of genes required for DNA synthesis, and it halts the cell cycle in the G1 phase. Upon phosphorylation by cyclin E CDK2, PRB releases E2F and enables the progression from G1 to S phase. Okay, so we're currently in here, and then here are all the different genes that must be activated to produce the correct proteins that will trigger these events. So in response to DNA damage, kinase ATM and ATR activate P53, which includes a P21 production. P21 inhibits cyclin E CDK2, maintaining PRB in its unphosphorylated state and bound to E2F, thereby arresting or stopping the cell cycle in G1. This arrest will allow time for DNA repair, or if repair is unfeasible, it will initiate or direct apoptosis to happen to prevent the propagation of damaged DNA. Right, so I'll be going over the mechanisms starting here, mechanism of cell cycle regulation, where CDKs bind to cyclins, and then phosphorylation and dephosphorylation take place. Inhibitory proteins come in, and then proteolysis will take place. So I'll start here, and then I'll be moving my way up from the CDK, where it gets bound to the cyclin, working my way around this diagram. So cyclins and CDKs collaborate very intricately to regulate cell division. Initially, in the M phase, CDK is inactive as mitosis begins. Cyclin production will lead to the binding with CDK, and it forms this inactive complex. So here we have the binding of these two, and we have the inactive complex here. All right. Activation will happen through phosphorylation at 309 160, or 309 at position 160, which positions a blocking loop. Okay. A blocking loop and dephosphorylation of tyrosine 15, or tyrosine at position 15, which allows for ATP binding. Okay, so we're talking about this over here. I'm going to come over here. These modifications will allow they will allow the activation of cyclin CDK complex that needs to form. Once active, this CDK will further promote its own activation. Inactivation follows when CDK phosphorylates DBRP, tagging cyclin with ubiquitin. This ubiquitin tag will signal proteasomes to degrade cyclin, thereby halting the CDK activity and completing the cycle. All right, so disruptions in the cell cycle can lead to various diseases, including cancer, and due to several key factors. So uncontrolled cell division can also occur in tumor formation as cells proliferate without the usual checks and balances. So whenever we have too little death, all right, cell cycle proliferation happens because cells that should be programmed to die are not dying. And then here we just have cell proliferation on blast, where cells are just continually dividing and dividing rapidly. And these are some of the diseases that form. Okay. I'll be creating an example here shortly. So cell cycle dysregulation significantly contributes to excuse me, colorectal cancer, which is the third most common cancer and second leading cause of cancer related to deaths globally. Despite advances in understanding and screening for colorectal cancer, the incidence continues to rise. Dysregulation in several key signaling pathways excuse me, play a crucial role in the development of this colorectal cancer. These pathways are integral to cell cycle control, and when disrupted can lead to unchecked or uncontrolled cell division. Additionally, changes in methylation patterns impact various processes, including cell cycle regulation, transcription, apoptosis, angiogenesis, invasion, and metastasis, further driving cancer progression. Understanding these mechanisms is so important for the development of targeted therapies and for improving colorectal cancer management overall. So, to conclude my presentation, proper cell cycle regulation is crucial. It is imperative for health reasons. Disruptions in the cell cycle can lead to serious diseases. As we know, the most common form of uncontrolled cell division is what we know as cancer. And here in this picture, in figure 1, it's an illustration of what unregulated cell division looks like in one colon cell that resulted in primary cancer, which subsequently metastasized to the liver. Secondary cancers appear as white patches. So you can see some of these white patches here in this picture. All right. So this sample was also obtained from an autopsy. All right. These are my references. Thank you.